In this study, we demonstrated a novel function of GLP-1 receptor agonists in protecting diabetic retinopathy. One meta-analysis of 6 large trials suggested that GLP-1 receptor agonists did not protect DR. Another meta-analysis of 93 trials revealed favorable effects of GLP-1 receptor agonists on late-stage DR compared to insulin. To evaluate GLP-1 receptor agonist effects, we conducted a retrospective cohort study of type 2 diabetic patients in our hospital. After step-by-step exclusion, 1626 patients were included in this study and were divided into 3 comparing groups according to their medication history. Utilization of GLP-1 receptor agonists significantly reduced their incidence compared to oral anti-diabetic drugs and insulin. We evaluated GLP-1 receptor levels and found that GLP-1 receptor was highly expressed by retinal vessels, which was downregulated in diabetic retinas, suggesting that GLP-1 signaling was compromised by diabetes. To testify the effect of GLP-1 receptor agonists on DR, we treated 2 cohorts of diabetic mice with semaglutide and losartanide, respectively. Electroretinogram results show that GLP-1 receptor agonists restore the amplitudes of scotopic A wave and B wave. OCT measurement further revealed that GLP-1 receptor agonists preserve retinal thickness. Immunostaining of GFAP confirmed attenuated inflammation. Consistently, diabetes-induced retinovascular tortuosity was suppressed by GLP-1 receptor agonist. We further examined acellular non-perfused vessels by co-staining collagen 4 and IB4. Results show that GLP-1 receptor agonists reduce the number of acellular vessels. Immunostaining of STING and phospho-TPK1 both indicated that STING signaling was activated in diabetic retinal vessels. Both treatments of GLP-1 receptor agonists potently downregulated the levels of STING and phospho-TPK1 in the vessels. The CIEP mediate the protective effects of GLP-1 receptor agonists in DR. First of all, we examine the status of CIEP in HRVCs, a human retinal vascular endothelial cell line. PA induced the levels and phosphorylation of CIEP, whereas treatment of exendin-4 counteracted this induction, indicating an inhibitory effect of GLP-1 receptor agonists on CIEP. A CIEP inhibitor blunted exendin-4's suppressing effect on STING activation and neutralized its protection on endothelial tight junction. In summary, the current study demonstrated that the GLP-1 receptor is enriched in retinal vessels, which is downregulated by diabetes. GLP-1 receptor agonists suppress the diabetes-induced activation of STING signaling, both in retinal vessels and retinal vascular endothelial cells. CIEP mediates the protective effects of GLP-1 receptor agonists on retinal vasculature under diabetic conditions.